Hi everyone, we will continue exploring the topic Bioinformatics. In this session, we will learn about a protein-ligand binding affinity model. This presentation is based largely on the information provided on the book Deep Learning for Life Sciences and an individual's Kaggle post explanation of the MolNet. In our last session, we explored the DeepChem library to download a pre-processed core dataset containing essential components, core proteins, ligands, and their complexes. We successfully extracted relevant information and visualized the ligand protein and protein-ligand complex using the AngleView.show_mdTras function. Building on this foundation, we will leverage the same core dataset to develop a machine learning-based prediction model for binding affinity using DeepChem. In session 7, we explored the information contained in the PDB bind dataset, specifically in the compressed file pdb-bind_core_df.csv.gz. Today we will use the load_pdb-bind function. My understanding is that the function uses three key components from the PDB-bind dataset: protein_pdb, protein structure data; ligand_pdb, ligand structure data; and the label, which is the binding affinity values. To study the binding behavior between a protein and a small molecule, we first need to featurize the protein-ligand complex. Why is featurization necessary? Featurization transforms the raw training data into a format suitable for machine learning or deep learning algorithms. For these computational models to learn effectively, we must represent the protein-ligand complex in a way that the computer can interpret. Then, can we use featurization techniques designed for small molecules, such as structural keys or other two-dimensional representations? The answer is no. As we discussed, protein selectivity is largely determined by its three-dimensional structure. Breaking it down into small fragments removes its structural content and consequently its functional relevance. In other words, applying traditional small molecule featurization method to proteins eliminates critical information needed for accurate predictions. Therefore, the key takeaway is that the two-dimensional featurization technique alone misses essential spatial and structural details, making them unsuitable for capturing the true binding behavior of protein-ligand interactions. To better capture the intricate interactions in protein-ligand systems, we turn to more sophisticated featurization techniques. Grid featurization is specifically designed for datasets like PDB-bind, incorporating both ligand and protein structural information. Developing a featurization algorithm for protein-ligand interactions is non-trivial. It requires deep chemical knowledge to extract meaningful features. Then, how does it work? This technique explicitly scans the three-dimensional structure for critical physical interactions such as hydrogen bond, salt bridge, and hydrophobic interactions. It leverages well-established principles of protein physics, ensuring that important known interactions are accounted for. Since it relies on existing physical knowledge. It may overlook novel interactions that are not yet well understood. In DeepChem, the RDKitGridFeaturizer simplifies this process by summarizing relevant chemical information into a compact vector, making it suitable for learning algorithms. Atomic featurization takes a different approach by representing the three-dimensional positions and identities of all atoms in the system. Instead of relying on predefined features, the learning model discovers patterns of its own, identifying new interactions and behaviors that may not be explicitly known. The advantage is that it allows the machine learning model to uncover previously unknown molecular interactions. However, this approach is computationally expensive, requiring significant resources to process and analyze large datasets. Both grid and atomic featurization offer unique strengths. Grid featurization ensures we capture well-known interactions, while atomic featurization provides the flexibility to discover new ones. The choice depends on the balance between the computational efficiency and the depths of interaction discovery needed for a given study. Then, where can we find binding information for protein-ligand complex? To train machine learning or deep learning models on protein-ligand interactions, we need access to high-quality data sets. In the DeepChem module, there is a comprehensive dataset collection called MoleNet, MoleculeNet. MoleculeNet is a collection of datasets within DeepChem specifically designed for machine learning and deep learning applications in chemistry and biology. These datasets can be accessed using functions in the dc.molenet submodules. As of today, MoleculeNet contains 42 datasets covering a wide range of applications, including structural biology datasets like PDB-Bind, which provide viable binding information. Here is an overview of 42 datasets categorized by their specific applications: quantum mechanical, structural biology, microscopy, material properties, physical chemistry, molecular catalog, chemical reaction, biochemical, biophysical, and physiology. PDB-Bind dataset is under structural biology. The overall modeling approach remains similar to what we have explored in previous sessions. However, our focus today is on one specific biophysical featurization technique, grid featurization. Let's dive into an example model. First, we import the DeepChem module using the alias DC. Next, we create a descriptor instance for grid featurization using the function RDKitGridFeaturizer. This function transforms protein-ligand complexes into a voxelized three-dimensional representation, allowing machine learning model to learn from spatial interactions. This particular function takes several key arguments. First, sanitize equals to true, automatically fixes errors detected by DeepChem. Flatten equals true, outputs a one-dimensional feature vector for each input. Voxel_width equals 2.0 defines the voxel grid size as 2 angstrom. A voxel is the three-dimensional equivalent of a pixel. Feature type specifies which interactions to search for in the three-dimensional structure, including hydrogen bond, salt bridge, pi stacking, cation-pi interactions. Additionally, RDKitFeaturizer computes two types of fingerprints: extended connectivity fingerprints, structural protein-ligand interaction fingerprint. So in summary, this particular function includes various arguments. The molecule net dataset collection within DeepChem provides PDB bind, which contains structure and binding affinity data. We load this data using the function load_PDB bind, which uses several arguments. First, featurizer equals to grid applies grid featurization during data loading. We have descriptor instance here. This goes to here as an argument. Splitter equals to random. Randomly splits the dataset into training, validation, and test datasets. Subset equals to core. Uses only the core dataset for modeling. We have seen the core dataset in the previous examples. Now the splitting the dataset for machine learning. This particular dataset returns as a result of this function gets split into training dataset, validation dataset, and test dataset. Then we use random forest regressor, an ensemble learning method, as our model. We import the random forest regressor module. We create the descriptor object sklearn_model. Then we create a prediction model with the descriptor object using the function sklearn_model. Then we train the model with the trained dataset with the function fit. Finally, we evaluate the model using the Pearson's R-square score. We're evaluating using the evaluate function with the trained dataset. We're evaluating the Pearson's R-square score with the test dataset. Results will look similar to this. The training dataset R-square value is 0.8716 and Pearson score for test dataset is 0.08634. When we look into the training dataset further, it has X values and Y values. Y value is the binding affinity and X values are the features, and the weight vector is also shown. The weight is mostly just 1 for the first 5 protein-ligand complex. I'm showing you the X, Y, and weight data for the first 5 protein-ligand complex. This is to see how the data looks like for a good runs. If you try to execute the code now, you will get the Pearson's R-square value of nan and nan for some reason. When I print out some X dataset in the training_dataset, I noticed that all X values were two-dimensional but all 0 for some reason, and thus the machine learning model could not build the proper model. Let's run the same model in the Google Colab environment. First, we install DeepChem and we import DeepChem module. We build a descriptor object grid_featurizer and we load the data with the grid featurizer. We split the data into train, validation, and test dataset, and we import random forest regressor module, and then we build our model with the random forest regressor function. and we train the model with the trained dataset. Then we evaluate the model with the Pearson's R-square value for the trained dataset and test dataset. We print out the result here, and then for the verification reason, we try to see what's wrong with this dataset by printing out the trained dataset for X value and Y value and weight values. Okay, let's run the code one by one cell by one cells. Okay, this part runs fine, and we import the DeepChem module. Okay, successfully imported DeepChem module, and then we create the descriptor object with the RDKitGridFeaturization function. Okay, and then we load the dataset with the GridFeaturizer. As you can see that there is something wrong with the dataset. The array is empty, and there should be 197 dataset, 192 dataset here, but it seems that the dataset is empty. Okay, we continue on running, splitting the dataset, and then we import the random forest regressor. Then we create the model and train the model with the random forest regressor and fit function. Then we evaluate the data. Then we print out the results. As you can see that there's no value for the Pearson's R-square for the trained dataset and test dataset. We look at the XY data and the weight data, the shape of the dataset. Okay, 154 dataset, trained dataset. Okay, this is two-dimensional data and this is one-dimensional data. When printing out the first 5 X, Y, and weight data, you can see that the X data is all 0, but there are some Y values and weight value for the first 5 data in the train underscore dataset. These results are consistent with my other results in the presentation slide. I believe that they might have changed the PDB bind dataset in the MONAD, or they might have changed the source code of the function since my last successful run. This is the end of the session. In the next session, we will try to build the binding affinity model differently with more in-depth knowledge of the model and PDB bind dataset. Thank you for your attention. I hope you found the session engaging and informative.